Respiration is defined as the controlled release of energy from organic compounds and cells to form ATP and proceeds as detailed in the following formula. The first stage of cellular respiration is glycolysis. During glycolysis, or sugar splitting, glucose is split into 2 to 3 carbon sugars and then oxidized, and the remaining atoms are rearranged to form 2 molecules of pyruvate. Glycolysis occurs in the cytoplasm and oxygen is not required, making it an anaerobic process. Glycolysis is split into 2 different phases, the energy investment phase and the energy payoff phase. Energy investment stage, ATP is used to phosphorylate glucose compounds. 2 ATP molecules are used to split a 6-carbon molecule. Hexokinase transfers a phosphate group from ATP to glucose, and the glucose is converted to fructose 6-phosphate. Then, a second molecule of ATP is invested as phosphofructokinase transfers a phosphate group from the invested ATP molecule to the opposite end of the sugar. The sugar molecule is then split into 2 3-carbon molecules of glyceraldehyde 3-phosphate. These molecules then enter the energy payoff phase. In stage 2 of glycolysis, the energy payoff phase, glyceraldehyde 3-phosphate is oxidized by the transfer of electrons to NAD+, and NADH is formed. Subsequently, the energy from this exergonic reaction is used to attach a phosphate group to the oxidized substrate. Then, 2 molecules of ATP are made via substrate-level phosphorylation. Phosphoglyceromutase then relocates the remaining phosphate group and a double bond is formed in the substrate as a water molecule is extracted, resulting in phosphoenol pyruvate. The phosphate group is then transferred from phosphoenol pyruvate to ADP, which is converted to ATP via substrate-level phosphorylation.
Step 2. Pyruvate oxidation. Pyruvate oxidation links glycolysis and the Krebs cycle. Pyruvate is first converted to a compound called acetyl coenzyme A before entering the mitochondria via active transport. The linking process links glycolysis and the citric acid cycle. It is carried out by a multi-enzyme complex that catalyzes 3 reactions. First, pyruvate's carboxyl group becomes fully oxidized and given off as a molecule of carbon dioxide. Next, the remaining fragment is oxidized and the electrons transferred to NAD+, which stores energy in the form of NADH. Finally, the coenzyme A attaches via its sulfur atom to the 2-carbon intermediate forming acetyl-CoA. Acetyl-CoA has a high energy potential which is used to transfer the acetyl group to a molecule in the citric acid cycle, a reaction that is highly exergonic. Citric acid, or the Krebs cycle, functions as a metabolic furnace that further oxidizes organic fuel derived from pyruvate. The cycle generates 1 ATP per turn by substrate-level phosphorylation, but most of the chemical energy is transferred to NAD+ and FAD during the redox reactions. For cellular respiration, the Krebs cycle occurs twice for each molecule of glucose that enters cellular respiration. One cycle of the Krebs cycle produces 1 ATP molecule, 3 NADH molecules, 1 FADH molecule, and 2 carbon dioxide molecules. Acetyl-CoA from oxidation of pyruvate adds its 2-carbon acetyl group to oxoacetate producing citrate. Then citrate is converted to its isomer isocitrate by removal of 1 water molecule and an addition of another. Next, isocitrate is oxidized, reducing NAD+ to NADH. Then, the resulting compound loses a carbon dioxide molecule. Another carbon dioxide molecule is lost, and the resulting compound is oxidized, reducing NADH to NADH. The remaining molecule is then attached to coenzyme A by an unstable bond. Next, coenzyme A is displaced by a phosphate group, which is transferred to GDP, forming GTP, a molecule with a function similar to ATP. GTP can also be used to generate ATP. Then, 2 hydrogens are transferred to FAD, forming FADH2 and oxidizing succinate. Addition of a water molecule rearranges bonds in the substrate. And lastly, the substrate is oxidized, reducing NAD+ to NADH and regenerating oxoacetate.
The next step in the process is oxidative phosphorylation, the electron transport chain. The electron transport chain section of cellular respiration takes place in the mitochondria's inner membrane. The electron transport chain itself is made up of 4 protein complexes, a small cytochrome protein, and an organic molecule called ubiquinone. FADH2 and NADH from the previous stages of cellular respiration bring electrons to the electron transport chain. The energy of electrons is converted to power ATP synthesis. The electrons received from the electron carriers, NADH and FADH2, are moved through a series of redox reactions. With each redox exchange, free energy is lost. The free energy released as the electrons move down the transport chain is released in small amounts to be used to make ATP. Oxygen is the final electron acceptor. Once an oxygen atom has gained 2 electrons, it picks up 2 protons from the mitochondrial solution and becomes water. The released energy is then used to pump hydrogen ions from the matrix across the inner membrane.
The last step of cellular respiration is chemiosmosis. This is the process in which energy stored in the form of hydrogen ion gradient across a membrane is used to drive cellular work such as ATP. The power source for ATP synthesis is a difference in the concentration of H+ on opposite sides of the inner mitochondrial membrane. In the mitochondria, the energy for gradient formation comes from exergonic redox reaction along the electron transport chain, and ATP synthesis is the work performed. Now for the steps. The first step is that H+ ions flowing down their gradient enter a channel in a stator, which is anchored in the membrane. Next, H+ ions enter binding sites within a router, changing the shape of each subunit so that the rotor breaks or spins within the membrane. Next, each H+ ion makes one complete turn before leaving the rotor and passing through the second channel in the stator into the mitochondrial matrix. Then, spinning of the rotor causes an internal rod to spin as well. This rod extends like a stalk into the knob below it, which is held stationary by part of the stator. Then, turning of the rod activates catalytic sites in the knob that produce ATP from ADP and PI. Lastly, depending on the cell, about 26 to 28 ATP molecules are formed, and the entire process of cellular respiration produces about 30 to 32 ATP molecules.